Hello everyone, in this video we're going to talk about midterm review part 7 where we talk about cells, cell organelle, and cell transport. So we're going to start with the first question, what is the basic unit of life? We're talking about cells. Those are the basic unit of life. When you are looking at a cell, you can still call it a life. If you're looking at a specific organelle inside the cell that's much smaller than a cell, then you would not call that a living thing. Are all cells the same size and have the same function? Obviously no, right? In your bodies, for example, you have neurons that are really, really long and pretty small. You have red blood cells that are completely different looking from neurons, right? So not all cells are the same size and they definitely have different functions. What does it mean when we say multicellular versus unicellular? If you have an organism that has multiple cells, then that would be a multicellular organism, such as us. And then we also have something called a unicellular, meaning that this one organism is only consists of one cell. What type of organisms are always unicellular? Those will be the prokaryotes. When we talk about cells, we need to think about how did people find out about cells. That's when we started learning about microscopes, right? Microscopes were in charge of the discovery and the learning of cells. The three parts of the cell theory were made when we learned more about cells as well in the history. So here are the three parts of the cell theory. We have all living things are made up of cells. Cells are the smallest unit and structure of life, right? Cells are makes up life. And then all cells come from pre-existing cells as well. So cells can't come out of nowhere. It has to come from pre-existing cells. Then let's talk about a few questions on microscope. How does a light microscope magnify an image? It uses light, right? So the light shines through the image, the cell that you're looking at, and then you can see what's inside the cell. An electron microscope, however, uses electrons. So we shoot electrons at the cell, and then that will allow us to form an image. The type of microscope that has a higher resolution and a higher magnifying power will be the electron microscopes. The type of microscope that you can use to see cells that are alive are only the light microscope. During the process of making a slide specifically for the electron microscope will not ensure the survival of the cell. So when you're using an electron microscope, one downside is that the image that you see is a still image. The cell is no longer alive. Can you see color or stain of the specimen when using an electron microscope? The answer is also no. What are some pros and cons of using a light microscope? So again, we said a light microscope, you can see the colors, you can see a live cell. However, the magnifying power is not as high as an electron microscope. Electron microscope is the complete opposite. You can't see a live message, that's the con, and you can only see black and white image, but you can see a much more magnified image. Difference between a scanning electron microscope and transmission. Scanning electron microscope scans the surface of the cell, so you can see, everything that's going on the surface, the very outside of the cell. But transmission electron microscope, what it does is that you take a slice, a very thin slice of the cell itself, and you get to see all the organelles inside the cell. What type of microscope do you use to see the three-dimensional cell surface? That will be the scanning electron microscope. All right, moving on, talking about two different big categories of cells. There are the prokaryotes, or organisms. There are prokaryotes and eukaryotes, and prokaryotes, again, are always unicellular. But there are some fundamental differences between the two types of organisms. Starting with the first one, what structures are common for both? So the cell membrane, the DNA, ribosome, and cytoplasm. Those are all common for all of the prokaryotes and the eukaryotes, okay? And then we can look at more questions a little bit more in detail. So we have, does it have a nucleus? What makes a prokaryote is that it doesn't have a nucleus. That's one of the most significant difference between the two. Eukaryotes do have a nucleus. Is it small or big? Prokaryotes are a lot smaller, and the eukaryote is much bigger. A prokaryotic cell is more complex, and a eukaryotic cell is less complex. A eukaryotic cell is more complex. Does it have a cell membrane? Yes and yes for both. So every single cell needs a cell membrane. Does it have a cell wall? So for prokaryotes, they all have cell wall. For eukaryotes, you would say sometimes. Because if you have a plant cell, then it does have a cell wall, and animal cells do not have cell walls. Does it have a DNA? Yes and yes, because all cells need certain genetic information that allows the cell to function, right? To pass on the message to the next generation. So everything has DNA. The shape of the genetic material. Prokaryotic cells have a circular DNA, and eukaryotic cells have a linear DNA. So a linear is a little string, a pretty long string, and a prokaryotic cell, you have a circle for a DNA. Does it have a membrane-bound organelle? So membrane-bound organelle means the organelles itself, some of the organelles are made out of membranes, the same type of membrane that the cell membrane is made out of. Prokaryotes do not have membrane-bound organelles. So for example, Golgi body, the ribosome, no, not the ribosome. Ribosome is not membrane-bound. The chloroplast, mitochondria, those are all membrane-bound organelles, and those don't exist for prokaryotic cells. Eukaryotic cells do have membrane-bound organelle. What type of organisms have this type of cell? For prokaryotic cell, we have bacteria and archaea, and for eukaryotic cell is plants and animals. There are other ones, but you can just remember plants and animals. Now we're going to take a look at some more details about cells. We're going to look at the organelles of the cell. Okay, so here's a picture of a cell. You need to be able to point out what each part of the cell is if you're given a picture. So let's start with the first question. When we talk about the nucleus, that is our control center of the cell. Here's our nucleus. The nucleus has a structure called this chromatin or chromosome. So how it works is that the chromatin is kind of this messy fiber of DNA wrapping around histone. So histone are protein that DNA can wrap around in eukaryotic cells, so to keep the DNA more organized. So that's a chromatin. Chromosome is the condensed version of chromatin, right? So if you're wrapping around DNA, wrapping around histone so tightly that it becomes super condensed or super coiled, then you form a structure called chromosome. Histone is the protein that DNA wraps around. A gene is a piece of DNA that codes for a protein or a trait. The nuclear envelope is the outside membrane of the nucleus. It's made out of the similar material as the cell membrane itself, so it keeps everything inside the nucleus for eukaryotic cells. We're right now just talking about eukaryotic cell. Nuclear pore are these channels, these protein channels on the outside membrane, on the nuclear envelope. So these allow a certain material to go in and out of the nucleus. And then we have the nucleolus, which is the very condensed region of the nucleus. Moving on to cytoplasm, that's the liquid jelly-like gel inside the cell that allows a lot of material to float around and travel from one place to the next to allow a lot of reactions to happen. So that's the cytoplasm. The cell membrane, the plasma membrane, they're the same thing, cell membrane, plasma membrane. The function, it keeps everything, inside the cell when it's supposed to be inside the cell and keeps everything out, and it allows certain waste and nutrient to go in and out of the cell. It also, allows cell transport, right? Material can go in and out of cell. We call it the fluid mosaic model. So I'll write this one down for you. Fluid mosaic model. So we call it fluid because the cell membrane is not a solid structure that never moves around. It's kind of like if you were to blow a bubble. You can see that, you can see the outside bubble, parts of the bubble kind of moves around. So is the plasma membrane. It's a fluid structure. Everything on the cell membrane kind of moves around a little bit. What are the components of the cell membrane? There are three parts that you need to remember. The cell membrane is mostly made out of something called phospholipid, which is a type of lipid. All you see over here is head and tail. Those are all phospholipids, okay? And then there are two other minor, not a little bit minor compared to the plasma membrane, compared to the phospholipid. So we also have proteins that are embedded inside the cell membrane, and you have some carbohydrates sometimes attached directly to the phospholipid or to these proteins. So there are three parts to the plasma membrane. Again, we have phospholipid, we have proteins, and we have carbohydrates. Moving on to mitochondria. So if you already watched yesterday's video, you know how mitochondria works. The only thing I want to point out is that what is the theory of endosymbiosis? So we say that mitochondria, from our observations as scientists, we think that mitochondria and chloroplasts both came from prokaryotes. They were originally prokaryotes that were able to do the job that mitochondria and chloroplasts do. So what happens is that there was an ancient, larger prokaryote that kind of took in smaller prokaryotes and became a part of it. And there was this symbiotic relationship between this chloroplast-like bacteria and this mitochondria-like bacteria. And then eventually these structures became a part of the cell, and then now we have eukaryotic cells that has more complex structures that includes the mitochondria and the chloroplast. So that's the theory of endosymbiosis. Endo means inside. Symbiosis is this mutually beneficial relationship, right? So that's where we think mitochondria came from in the first place. The next one is called ribosome. So ribosomes make proteins, which are very important. Proteins make up a lot of structures. Proteins are in the cell membrane to allow membrane transport. So proteins are very important. So we have these ribosomes making proteins using something called RNA. And RNA are made from DNA. Both DNA and RNA, as a reminder, are examples of nucleic acid. So that's the function. Ribosome has two components. Ribosome is actually made out of protein itself, but it also has a piece of RNA inside of it. So ribosome is a combination between protein and RNA. There are two types of ribosomes that you can find. One is called, they're ribosomes that are living on the rough yard. They're a part of the rough yard. That's what makes rough yard rough. So as you can see, there are these ribosomes attached to the rough yard. There are also ribosomes that are what we call free-floating ribosomes. So they're just in the cytoplasm, floating around making proteins for the cell itself. And then we have something called the rough yard. The rough yard makes proteins. Then we have the rough yard that makes proteins. But in comparison, there's also something called smooth yard. So the smooth yard has nothing to do with proteins at all. Smooth yard detoxified the cell. All right. So what it means is that if someone were to be poisoned for some reason, or if you drink alcohol, which is poisonous to your body, the smooth yard break that down so that it's no longer poisonous for your cell. And then we have something called Golgi. The Golgi body packages and transports protein. So what happens is, if you look right here, we have proteins that can be made in the rough ER, and then these proteins, if it needs to go out of the cell, it wouldn't know where to go, right? So what happens is that these proteins will be packaged into a little vesicle, a little bubble. And then this vesicle will travel to the Golgi body. And then the Golgi body will kind of put markers on this protein to tell it where it's going. Okay, so it's kind of the address for your envelope, telling you where to go. That's what Golgi does. It packages and helps transporting proteins. Vacuole is a structure that is common, is mostly in plants. And for plants, you have this large central vacuole, this large water tower almost, that stores water for the plant. And then we have vesicles that are in charge of transporting material, basically. We have lysosome that helps breaking down material that is no longer needed in the cell usually. So let's say you have a protein that's no longer needed, or if your cell is digesting food, we can send the food to the lysosome and then it breaks down the food. It has digestive enzymes, digestive enzyme is good enough for you to know. So these digestive enzymes break down the material for the cell. What type of cell has a lot of lysosome? Liver cells have a lot of lysosomes because your liver helps detoxify your body. And then we're moving on to cell wall, a structure that only plant cells have. The cell wall keeps the structure of the plant cell, all right? And it also, think about this, plants can absorb water and lose water all the time, but you don't want the plant cell to shrink and expand too much, right? So the cell wall kind of holds the shape of the plant cell, and then whether there's too much water or too little water. What type of carbohydrate make up the plant cell wall? This is called cellulose. Let me spell it out for you. That's how you spell it. Cellulose is a type of carbohydrate that's the component of the cell wall. This one we already done. Flagella is a long tail behind a cell, and this job is to help the cell move around. And then we have cilia, which are little hair-like structures all around the cell. You can draw it like this. Right? So that's cilia, and how cilia does is that does its job is that all the cilia will float in one end, and then it will push back to push the cell to another direction. And then we have pseudopod, which is also in charge of helping the cell moving around. So what you see over here, this eukaryotic cell can extend a part of its cell membrane in a certain direction. Let's say it's trying to eat this food, right? And then it'll bring its entire body of the cell over to surround the food and then eat it. Cytoskeleton is kind of like our skeleton. It helps holding the shape of the cell. And it allows certain material to travel inside the cell as well. Differences and similarities between plant and animal cell wall. It's important for you to know that chloroplasts are only for plant cells, and cell wall is only for plant cells as well. The difference between Golgi and ER, again, ER makes protein and Golgi processes and helps transporting proteins. Where are they located? How do you know? All right, let's look at this picture. So rough ER and the smooth ER are both directly attached to the nuclear membrane, so it's right outside the nucleus. And then we have the Golgi apparatus, which is a little bit farther away from the nucleus. It's floating on its own. All right, moving on to cell transport. Cell transport, what are the two components of a solution? Solution is made out of solute and solvent. What is diffusion? Diffusion is any kind of molecule going from high to low of concentration, high concentration, area of high concentration of this molecule to an area of low concentration of this molecule without the use of energy, right? This is really important, both the high to low part and the without the use of energy part, right? So if you look at this right here, we have these little magnets, right? We have these green magnets, and then we have this very concentrated region of the green magnets. So what happens is that molecules are constantly moving. They'll bump into each other, right? So as they bump into each other, they get more spread out, right? Because they keep bumping into each other, right? So then the molecules are moving from an area of high concentration over there to this area over here that's lower concentration. That is called diffusion. Anytime you have this, it's called diffusion. All right. The next question is diffusion. During diffusion, right, molecules go from high concentration to low concentration. It does not require any input of energy. The effect of temperature is that the higher the temperature, the faster the diffusion rate, right? Think about this. If you were to make tea, it's hard to make tea using cold water because whatever is inside the tea bag is hard to diffuse out of the tea bag. But if you use hot water, it'll make the process a lot easier. What type of molecules can travel through the cell membrane using simple diffusion? Those are small and nonpolar molecules. So what happens is the cell membrane is made out of mostly phospholipids, and they look like this. Right? They have these head and tail regions. The heads are what we call hydrophilic over here as well. And then we have these tail region, which is called hydrophobic, right? So these are the tail region is hydrophobic. So if you have a molecule that's really large, such as this one, it's not that large. It's a larger one. This one is hard to get through the cell membrane because there's not enough space in between the phospholipid. So it's much more helpful to have small molecules, right? Simple diffusion means we're not using any tools at all. We're just having molecules going through the cell membrane directly. So small molecule is nice. Nonpolar molecule is important because if you have a polar molecule, it's hard to travel through this large region of nonpolar region of the cell membrane, right? So if you have a nonpolar molecule, it can go its entire way to the other side of the cell membrane. Equilibrium means equal concentration. So if you have the cell membrane and you have, this area this big on each side, and you have the same number of molecules on each side, then the concentration is the same. That is called equilibrium. Okay? Do molecules move across the membrane during equilibrium? The answer is yes. So during equilibrium, you have the same concentration, but molecules are going to move around no matter what. So, but the idea is, when one molecule moves through, another molecule would immediately go back, right? Because now you have high concentration. Molecules want to reach or tend to reach equilibrium. So now you have too much over here. This molecule will travel back. So you have this what we call dynamic equilibrium, where there's still movement of the molecules, but you have equal concentration on both sides. The other type of membrane transport is called facilitated diffusion. So if you have a molecule that is larger and or polar, so let's see, that molecule. So if you have a molecule that's large or polar or ionic, then it's difficult for it to travel through the cell membrane directly. So what would happen is that if a cell needs to get certain type of molecule in and out of the cell, it will build itself kind of a special door for this type of molecule. So this is called a membrane protein. So these membrane proteins have channels inside of it to allow the molecule that are large or nonpolar, or large or polar or ionic, to travel through the cell membrane, because these large or polar or ionic molecules cannot go through the cell membrane directly, right? And then, so facilitated diffusion is membrane transport from high to low concentration without the use of energy, but with the use of membrane proteins. So what type of molecule is required for facilitated diffusion? That will be proteins. Molecules traveling from an area of what concentration to what concentration during facilitated diffusion, again, it's still diffusion, and anytime you have diffusion, it's high to low. Right here, high to low. There we go. All right. In the next video, we'll discuss osmosis and everything after that, but hopefully you understood what was discussed in this video.